showing the first analysis of the data set, fully blinded analysis, of course. Thank you.
Thank you, David. So I'm Leihong from NCTR. As said, although I'm a colleague of Schroeder, I didn't know anything about the validation data set. So today I will introduce a deep learning framework in order to do this challenge. So here, since Wade and Schroeder have extensive introductory things about the DILI and about the data set, so I'll go through this part very quick. So I just show you from my perspective how I understand about this question from a machine learning guy. So first about the DILI, the only thing I know is DILI very important, and this is the endpoint of this challenge. So this is my task, you know. And for the CAMDA challenge, because when we don't have any restrictions on the modeling algorithm, and we don't have any restrictions on external knowledge. But to be fair, because I'm the in-house guy, you know, so I don't use any external knowledge. I'm only use knowledge, the information provided by the CAMDA challenge. So that's why we can compare it to the different teams to see can we get any advantages from the external knowledge. And then this is a summary of this data set. As Schroeder have introduced, we have 190 of them are provided as a training data set, which means their label information are known to me. And we have 2 different cell lines, and we have 2 different microarray platforms. So here is what I thought about a general modeling workflow. First, when you got the data, we might do some data pre-processing, and then the data will split into a train and a test part. So because our model was solely based on the train part, to evaluate our model unbiasedly, in most cases, I think people will further, you know, divide the train set into 2 different sets. That is the subtrain set and the sub-development set or sub-dev set. For the subtrain set, we are using it to train the model, to optimize the model parameters, and then we evaluate our model on the development set. Because the development set information are not used in the model construction, so it is kind of unbiased. And if you only, you know, separate the data once, it is called a holdout strategy. But if you separate them many times, it will call like the leave many out or cross validation. And particularly for this challenge, since we have 2 different cell lines, so we can also evaluate our model on another way. That is what I said here, the cross cell line validation, which means you build a model based on 1 cell line, for example, the PC3 cell line, and then evaluate your model performance on the MCF cell line. And finally, when your model was built, we will predict the holdout test set, the 86 drugs, and then submit it to the challenge. So let's talk a little bit about the data pre-processing. So first, you have to download the data from the SafeMap website, right? And for me, I use R package, the FE package, to extract the information from the cell file, and I normalize the data by MAS5 method. So you can also use other common methods like RMA. And then for the MAS5 normalization, I also further transform it to its log2 value, because it will be more normally distributed according to our previous study. And then since we have 2 different microarray platforms, I just simply merge them by their shared probes. There's 22,277 of them. And furthermore, I remove the control probes. There are 32 of them. And so finally, we will have 22,215 probes used as a raw feature set. And I use holdout strategy here. So I just randomly split the original 190 drugs into a subtrain and a sub-dev set with a 70 to 30% of the ratio. So which means we have 114 drugs used as a subtrain set. And then at the final step of the data pre-processing, I did the data scaling and normalization only based on the train set, which means I calculate the mean and the standard deviation value from the train set and apply them to normalize the dev set and later on the test set. So for the reproducibility purpose, I record all the steps of the data pre-processing in the R document, and they are now public online. And I also fixed the random seed, what I used to split the data set. So if you are interested in that, you can just go to this website, and I think you can repeat my result very easily by following my code. Okay, so let's go into the key part of this challenge, which is modeling. So, there's a lot of modeling algorithms can be used in such a challenge. And I just list a few of them. The random forest, the support vector machine, which are the most popular algorithms, I think, this decade. And we can also use the k-nearest neighbors, linear discriminant analysis, which is a very simple and intuitive algorithms. But as I mentioned in my title, I will use deep learning because, deep learning is very popular nowadays and have changed a lot of industries. And people, everyone, including me, trying to, combine myself to deep learning to get more audience, So for deep learning, so the simplest deep learning is what we call, what we said about deep learning. Mostly we are talking about a deep neural network. And the simplest form of the deep neural network is what I said is the multi-layer perception, which means you have multiple hidden layers between the input and output layers. So although this is, you can call this a deep neural network, but the most successful applications of deep learning are not use this such a simple way. They use a more complicated way, like the convolutional neural net, the recurrent neural net, to deal with some spatial information, the data with spatial information like images, text mining, voice recognition, something like that. And besides the supervised learning, people also are interested in the unsupervised learning, which means we don't know the endpoint, and we just train the network without the endpoint information. And we have another deep learning applications and model structures like semi-supervised learning, generative adversarial models, reinforcement learning. I'm not talking about them today. Today I will only talk about two very simple network structure, which is simplest deep learning, the multi-layer perception, and the unsupervised learning. So I will show you why I use these two. So first, because we are dealing with the gene expression data. So unfortunately, the features, we call this kind of data the tabular data, which means the features didn't have any orders or neighborhood relationships. So that means we don't have any spatial informations to run the convolutional neural net or to run the recurrent neural net. And but MLP, the simplest format of the deep learning, can handle any kind of the data format because it's fully connected. It can learn the structures by itself. Although it didn't have so many successful applications right now. And the reason I choose unsupervised learning is because we're dealing with biological data. So for biological data, we know there's a thing of the curse of dimensionality, which means we have too many features but not so many samples for normal biological data. So we will use the feature reduction to reduce the feature numbers. That means we can reduce the feature numbers, and our model will not that prone to the overfit. And also, the unsupervised learning is good at denoising. I will show you later. I will show you in a minute. And since the microarray data has a lot of people criticize about it for the noise in it, so that's why we use unsupervised learning here. And I just show you a very popular unsupervised learning. I think most of you already know about that, the autoencoder. So the autoencoder is a kind of compressing method, which consists of two parts. The encoding part and the decoding part. So the input, this is the image, but you can use any kinds of data as an input. We'll go through the encoding part to get to the bottleneck here, and this bottleneck have a low dimension than the input layers. So that means we can compress data from the high dimension to a low dimension for the feature reduction use. And then for the decoding part, the bottleneck will decode it to the original data. Here is the mushroom. And because when you use a whole autoencoder, we can reconstruct the input data to its output layer. So that means it always used as a representations. And as I shown here, because the network structure was fully trained by the input image or input data, so it is very data-specific representations. So back to our challenge, because we have two different cell lines, so when we use different cell lines as an input, I think the network will be quite different because they try to fit to represent the specific cell line data. And then another characteristics is the autoencoder is a lossy compression, which means it will lose some information of the input data. So, and because it is lossy, so it can be used for the denoising. So this example shown here is using MNIST dataset. So you can see we add some random Gaussian noises in the digital images, and after the autoencoder, we can train or we can reconstruct the image without these noises. So I think this is very important to us to dealing with some noisy data like microarray. And the most novelty comes to my framework is I use this autoencoder to handle multi-platform data. So let's go into the multi-platform data now. So now we are dealing with the connectivity map. So we have two different cell lines. So if you look at these two cell lines, we can find that they are sharing the same list of the drugs, and also because they are using the same microarray platform, so they also have the same list of the gene features. So which means they kind of are different batches, but you know they are from the different cell lines. So you cannot just average them, simply average them to get the final result. And you know they might involve in some platform-specific noises. And this situation is not just come from the CMAP. As Wada has talked about the OpenTG gates, they have the human rats in vitro data, the rat in vivo data, and most of them are applied on the same list of the drug, and also they are also using the same platform as microarray. And also we have the SEQC project. We have both microarray and RNA-seq for the same cohort, and we can find some mapping algorithms to map the microarray probes to the RNA-seq reads, so on and so forth. So how can we deal with the multi-platform data? So here, because we know the data, the information of different multi-platform from different platforms are highly paired. So they are not only sample-wise but also feature-wise paired. So we cannot just combine them via one single dimension. Like we can combine them via features. For example, we have 10,000 of microarray probes, maybe 10,000 of RNA-seq reads. We can combine them together to have 20,000 of features, but that will lose some information about the sample-wise pair. And also we cannot combine them via the sample dimension. So, and also we cannot use some pooling strategy because they are not completely equal. They involve some platform-specific noises, and these noises sometimes is even not independent to the sample variance. What is the endpoint? So we cannot use some very simple algorithm to deal with that. And the most common algorithm to deal with the multi-platform data is we will most of time we will build a consensus model, which means we build one model for each platform, and then we combine this model together to use some strategy like the winner vote, something like that, to get a final model. So a limitation of this kind of model is it is not very efficient. Let's say if you build a consensus model with 10 platforms, and then your model is very successful, and you want to apply it to a new test data. So when a new test sample comes, you suddenly find that you need all information from the 10 platforms. If you just miss the one platforms, your model might not be available, or at least your model's performance are not guaranteed because you are missing some information here. So by dealing with this challenge, so I read several papers from the deep learning, from the computer visions, and I was inspired by a work that was published in 2016. But I think in recent 2 years, a lot of papers are talking about them. It is called something like the domain separation networks. So let's just use an example from MNIST, which is like the golden standard data set in the image analysis. So here we have the digital images here, like from 0 to 4. If we want to build an autoencoder to represent this kind of image, the simplest way is we can build only one autoencoder. That is this arrow here. We build one autoencoder and we just extract the bottleneck to see whether it can represent this data set very well. So this is the simplest way. Or we can also build two autoencoders. So the left side is trying to capture the digital shape, as I shown here, the digital shape of this image, and the right autoencoder is trying to capture the background of this image. So you may ask, how can a machine or how can the algorithms know what is the digital shape or what is the background? The machine is not like human. He didn't know that, right? So that's why we need another data set. So this is an MNIST data set image, but with a background. So now you can see this. We also built two autoencoders. The left side is to capture the digital shape, but the right side is to capture the background. So now, because they all share the very similar information from the digital shape, so we will have a way to tell the machine how you can define which kind of information is the digital shape, because it is shared by two data sets. And this is what we call the shared content. And the right side about the background, because the background from this two data set, image data set was different, so this is so-called the private content. Okay, so let's going back to our gene expression profile. So I think the issue is the same. So now we have the final result of the expression value. So we know this expression value is influenced by three major factors. The first is influenced by the sample itself. So that's what we really need. So we wanted to see whether DILI drug have some high expressions on some certain genes, whether the non-DILI drugs are lower expressed. This is the sample inference. And the second one is the platform inference. That because we have two different microarray, two different cell lines. So different cell lines might also influence on the expression value. So some genes are not expressed in a particular cell line. And the third part is caused by the random error, which is the noise of the microarray platform or the noise of the technical part. So because we are using the autoencoder, we hope the autoencoder can help us to deal with the random error part, so which is the denoising. And then we are trying to use something similar to the domain separation network. So we're trying to separate these two different inferences or variances from each other. So in that case, you see the from image, we need a two data set. And fortunately, for the gene expression analysis, we have two different cell lines. So that's what I designed for this framework. So in general, I built three autoencoders. The first autoencoder is a very, ordinary one. So you just put one cell line as an input and also as an output. So this autoencoder is trying to represent this cell line, the PC3 cell line, for example. And the second autoencoder is trying to represent both cell lines. So you can see here, the input is a combination of PC3 and MCF7 cell lines. And of course, the PC3 is going to transform to PC3-2, and MCF7 is trying to transform to MCF7-2. And then I built a third autoencoder. So this is pretty tricky, and I'm still investigating that. So because the cell lines have some complicated intrinsic relationship between each other. So I trying to see whether we can build an autoencoder. Actually, it's not an autoencoder right now because the input and output is different. So to transform the PC3 to the MCF7, because we have the same drug list, we have the same features. So supposedly we can do this because they have the same dimension between these two data sets, and also vice versa, to transform MCF7 to the PC3. So then now we have 3 autoencoders, and then after that, after the autoencoder was trained, we use the raw input. I just said the 22,215 features to going through these 3 autoencoders and extract its bottlenecks, which I call the feature. The feature S means single, B means combined, C means cross, and then I just combine them together to get the intermediate feature set. Okay, so here after we get the intermediate feature set, we just simply flatten them into 3,000 features, and we're going through another deep neural net. This is a modular perception, and for the regularization, purpose, I add some dropout layers between each 2 densely connected layers, and the endpoint, the output layer or the endpoint is a DILI, label in this challenge. So here, let's say the performance. So because the different trained data. So if you use a PC3, you will have a different, the single autoencoder. But the autoencoder, the combined autoencoder and the cross autoencoder are still the same. But so we have the different training data set. We can use the MCF7 or we can use the PC3 to go through this process to train the autoencoder models. And finally, this is the performance. So we train the data based on 114 drugs, and then we test our model on the development set. So you can see the blue one is kind of a cross-platform validation, and the white one is, the in-platform prediction. So you can see the PC3 generally have a better performance for DILI prediction by our model in this challenge, but we are looking to see whether other groups have different observations than us. And also, because it's very interesting, because you can see the PC3 model have an equivalent performance on the MCF7 dataset. So, and it is even better than when you use the MCF7 to train the data. So that's why I will only use the PC3 to train the model and submit the predicting result on the test set later on. And first, we try to compare our performance to some regular machine learning algorithms like the random forest, k-nearest neighbors, support vector machines, LDA, the linear discriminant analysis. And we also found that based on our holdout strategy, this method cannot have didn't have any predictive power. But for the PC3 cell lines, all of them have a slightly positive MCC, but still our model showing the best performance right here. And because the first 4 models, the regular models, didn't use any information in the other cell lines, so it is not surprising to me that they cannot do any cross-cell line predictions. But since our model used these multi-platform informations to train the autoencoders, so you can see for the PC3 cell lines, we have good cross-cell line predictions, but for the MCF7, it didn't. It might be because the MCF7 itself didn't have a very good performance. Okay, so next I will compare. So because we use 3 autoencoders, and we try to see which autoencoder contributes more to the model performance. So I just build a model based on single autoencoder, or based on the combined autoencoder, or based on cross autoencoder, or based on the combination of any 2 of them, and finally compare them to the combination of 3. So you can see both the accuracy and MCC showing that when you combine 3 of them, it has a better performance, and also it has a better cross-platform prediction. Okay, so finally, I just submit this model to Strata and get the result back. So here we find our model. I only use as I said, I only use the PC3 model to predict both PC3 and MCF7 dataset because I believe my model can do some cross-platform predictions. So here you can see MCC here. It is better for the PC3 test set, but not that good for the MCF7 dataset. But if you're going back to see the MCF7 performance on the training dataset, you can see it is still similar to that performance, which means we are not lost too many accuracies or performance based on for our model when we transfer our model from the dev set, internal dev set to the external test set. Okay, so after that, after I was submit my model, and I got some suggestions from the CAMDA reviewers, and they're very helpful. And based on their suggestions, I further test some improvement of my model. That's quite trivial, but takes a lot of time. So first, I use the denoising autoencoder instead of the regular autoencoder, which means we have some dropout layers inside the autoencoders. And also we test other activation functions, like actually I test 10H, SALU, ReLU, something like this, to see whether it will have some influence on the model. And also we optimize the number of intermediate features, which means the features in the nodes in the bottleneck for each autoencoder models. So here's some results. So you can see here we applied the dropout, we applied the denoising autoencoders with a dropout ratio as 0.5. And we also changed our features, number of features from 1024 to 256. And we tested the cellular scaled exponential linear unit, which was published, I think, last year. And compared to the most common one, the value rectified linear unit. So if you, from the figure, we found the cellular didn't have a comparable performance and value, which means the order is better. I don't know. And we also extend our epochs from 500 to 100 to 2000. So you can see from here it have some fluctuated. It have some fluctuation because we have some dropout layers here, but still the performance in average was above like 0.25 in MCC, which was quite similar to what we have in the model we submit before. So the observations of this challenge is we found that the PC3 cell line is in overall has a better performance in DILI predictions, not just by our algorithm, but also by some other, like random forest and supported vector machine. And also we demonstrated that our model has the best performance compared to those, common modeling algorithms. And in addition, our model can also do some cross-platform predictions. So this is only my observations. I'm looking forward to the other groups. And the take-home message of this framework is that our proposed framework is specifically designed to handle multiple platform data. And I think it is promising to split the expression profile into a private content and into a shared content based on this kind of information. And because we are using this multi-platform information in the model training only in the autoencoder, so that means we don't need all the platform information for the test data. We only need one, the major platform, as I just showed you here, the major platform for the test sample. And we also demonstrate that autoencoder is capable to denoising and to represent the tabular features such as gene expression profiles. And although our proposed model here designed for this challenge is applied on multiple cell lines, but I think it may be also generalized to handle other types of multi-platform data, like comparing microarray and RNA-seq and other like human red in vitro. We don't have this investigation yet, but I think we will do that in future. And because this is a novel framework, so I'm pretty much sure that we need further improve the model structures, because current model structure is still very arbitrary by me, and we can improve the model structure in the future. And also we can test different normalization method for the microarray. And because for this analysis, I didn't use vehicle control samples, but maybe use these samples can further contribute to the performance. So okay, so that's all my presentation. Thank you for your attention. And I want to thank you, my team members, Joshua, Zhichao, Shrad, and Wade for their fully support and hard work. Thank you very much.
Thank you very much for this thorough presentation. We have time for one or two short questions. If if you want to come. I apologize, but maybe moving a bit is quite good after all that sitting.
Here you go, I'm back a bit later. Thanks for a very good talk. I just have 2 quick questions. So first one is, what is the computational cost for training this model?
We have HPC in our group, but the thing is, I don't think it will cost too much because we are not using GPU right now. We are using CPU. So when you transform it to GPU, I think it will only use maybe 1 week for the whole training process.
Right.
Yeah.
Okay. The next
it's
question.
affordable, right?
Yeah. The next question is related to the first one, actually. My second question is it worth filtering out some of the features before you conduct the training?
Yeah, that's a good question because actually this is a specific condition for the biological data, you know.
Right.
So to me, I don't prefer to fill out the feature because that will introduce some, you know, unpredicted outcomes. And I cannot compare my result to other audience. You know, if I say I only use 100 features for the whole training set, and you might think the feature selection will contribute to this a lot. Right. So that, because particularly for this challenge, I don't use this strategy you suggested. But I think, yeah, maybe later on we can improve our structures with your suggestions. Thank you.
All right. Thank you so much.
Thank you. Thank you. One more question? Okay.
So first, thanks for the great talk. My question is about from biological interpretation of the model you built. So for the features gene you used to build the model, are there really a biological related pathway there related to the liver injury?
Yeah, so that's a great question. So because we're using all the features. So basically we don't, because the intermediate features, as I said, the 3,000, you know, intermediate features, they're kind of combinations of the raw features. So they're not just raw features. We're not just extracting raw feature. We're doing some transformations on them. So I'm sorry, I don't think they have any biological meanings on that. And that is also, I just take a few more minutes because, okay, so because to introduce the biological information that will be helpful, but that will introduce another limitations by the current biological knowledge. So people might think, can we introduce like the pathways, the gene ontologies in our model to have a better structures so that they will have more biological meanings? But you can also understand that because the current knowledge base of the biological data set are not that complete. So if you based on this like pathway, you will restrict yourself on the pathway informations, and you will lose a bunch of informations because not all genes are included in the pathways. Only maybe let's just say 1,000 of them are included. That means 19,000 of them you are intermediate filled out because they don't have currently they don't have annotations. That's why I didn't use this kind of.
Yeah, from our side, we do want to understand the mechanism model. You said the 2 cell lines have different performance. So I think if we understand features, can give a better understanding why one cell line works better than the others.
Okay, so another way is we can see the feature contributions to the intermediate features. So, but we didn't do that yet. But I think we will take your suggestions. Thank you.
Thank you very much. So, in the interest of time.